So
Yes,
I think we can now go ahead with the last presentation on nanobodies.
Well, some fellow back there suggested that this is ghost presentation. I hope it's not. I came across nanobodies some 3 years ago. We are doing lipid membrane biosensors, and we are always trying new material just to put some ruggedness into the systems. Nanobodies are in the market for some time now, and a lot of research has been done around tumor detection, imaging, and drug delivery. First reports seemed very interesting to us because they presented nanobodies as yet another antibody fragment, but more versatile and more efficient than conventional antibody fragmentation products. The latest reports, however, there is a tendency to highlight the differences and the peculiar features that do not fit antibody-based models. Now you don't need me to say how well antibody technology has served us in developing clinical and environmental monitoring devices and detection kits. Antibodies are bulky molecules with a delicate nature, not always easy to handle, but engineering worked miracles to deliver us all sorts of fragments, yet at higher costs, lower affinities, quicker and largely unmanageable kinetics, and a tendency to aggregate. Heavy chain-only antibodies occur naturally and lack the light chains and a constant domain unit. They are more amenable to engineering and easily yield nanobodies and nanobody constructs like bispecific, tetramers, pentamers, or conjugates that are hydrophilic, soluble, stable, and quite robust. Nanobodies show a protruding complementarity determining region that penetrates otherwise inaccessible clefts and have some other features that could give a clear competitive advantage over conventional antibody technology once some problems are solved, such as the rapid renal and blood clearance, the lipophobicity that hinders membrane penetration, and production that yields a significant percentage of low affinity variants. Applications are plenty, that comes in, and in many cases progressed beyond proof of concept. Briefly, we have nanobodies against receptors, such as the epidermal growth factor or death receptors, and against various ligands, such as urokinase or chemokines, for delivering drugs, including Pseudomonas toxins, liposomes, micelles, or nanoparticles, for studying proteins, mainly intracellular, such as actins, catenins, and green fluorescent protein. Most of them have proven potential in vitro. There are clinical trials ongoing. Just the clinical trials for the drug targeting have been aborted recently. The main drawbacks seem to be poor membrane penetration and the fast clearance. And we have also for molecular imaging, both against targets or for labeling. This is the most successful trials yet. I skipped cancer biomarkers such as alpha-fetoprotein, prostate-specific membrane antigen, tumor-associated glycoprotein, and others. And last but not least, for my line of research, is for biosensors, for immunonanosensors. They have been developed mostly for toxins and biomarkers, and they have involved some high tech in our study area, surface plasmon resonance and sol gel technology. There is a main problem in immobilization. While biosensors, you have to put your biorecognition element on top of the transducer, so you need some immobilization technique. And nanobodies didn't seem to fit with the cross-linking and sol gel technology very well. The problem is orientation. And they have proven potential at bench scale, and research is definitely ongoing. Now, this is a very brief overview of the biosensor system. We are going to concentrate on this area. We have the biological recognition layer, which is immobilized on top of the transducer. What we are doing is using lipid membrane, bilayer lipid membrane, self-acceptant lipids on top of electrochemical transducer, or more recently, polymerizable lipids. And in fact, you can do anything with that area. We can put in antibodies and have an immunosensor, or we can put in enzymes and have a catalytic sensor, or we can use the lipid membrane bilayer itself as a recognition element. If we have different kinds of lipophilic compounds, we can play along with the lipophilicity indices and get, let's say, 2 signals. It's for one compound, or you can put in DNA and have a DNA sensor. You can put in receptors. You can put whole cells. You can do anything at bench scale, because lipid membrane biosensors have not been engineered to devices as yet. This is the main cell, the flow-through cell. We use graphene electrodes. It's simply graphene nanoseed, which is wrapped around copper, and we have the lipid membrane, which is polymerized on some filter, and we are just putting antibodies in, and we have the bioelectrode tip. What I'm going to show you is on saxitoxin detection. We're using anti-saxitoxin antibodies. So what we are doing, we do have them polymerized membrane. We just put it in the buffer solution. We have some stabilization. We just put in the antibodies, and we have the immobilization simply by physisorption. We put in saxitoxin, and then we have distinct signals depending on the concentration of the toxin. The more the concentration, the more the signal. The signal is completely reversible or transient signals. And what we're measuring is transmembrane ion current. And we do have a very nice linear semi-logarithmic plot that we can use to detect any unknown samples, and that's only to give you the comparison background for the nanobodies. So we do have here a detection limit which goes to 3 nanograms per mil. It's quite nice detection limit, but we wanted to reduce it. That's why we use nanobodies. We have a range that goes up to 70 nanograms per mil. We have a sensitivity of 60 millivolts per decade. Quite nice and narrow relative standard deviation, and quite a good reproducibility for between days and analyst detection. What we do need is to reduce the detection limit just below, and if possible, enlarge the range, keeping the sensitivity as is. Well, you're mostly used in human samples, but we're doing environmental samples as well, and environmental samples may be more complex than human samples. You don't know what to expect and at what range this would be. So we needed something that would optimize, as we say, the sensor. Some notes on that: we do use UV polymerization to construct the lipid film. Immobilization is based simply on physisorption. That leads to simple surface attachment with limited perturbation of the polar head region of the film. There is no concentration threshold, and that shows us that there is no membrane spanning. When we increase antibody loading, sensitivity increases, but there is a limit to it, because the aggregation of antigens, antibodies, and their complexes on the surface of the membrane, they block the membrane. And the mechanism of transduction involves changes of the surface potential and of dipole moment due to the antibody-antigen interaction. Response is pH sensitive, just a narrow range there, that permits sensor reversibility at another pH. Now, actually, heavy chain only antibodies were done in the bottom of our research, but I put it here because it makes sense. We just wanted to show you the linearity of the thing. Heavy chain only antibodies could be immobilized during polymerization. So we doing that, we have increased the antibody loading by 20%. Detection limit decreased by 12% and sensitivity increased by 8.5%. That's not big news for us anyway. The relative standard deviation decreased, and that was good news. And the precision, the reproducibility between days and analysts also got below 5%. The range decreased slightly. Again, there was no concentration threshold, and the mechanism of reduction seems to rely more on dipole moment perturbations. That gives more quick responses, and that make us believe that we have certainly lesser surface potential changes. This could be due to reduced steric effects at the membrane surface, since the size of the biorecognition species is smaller and less complicated, and the response still remains pH sensitive. Now, the nanobodies, the antibody mobilization, there are more ragged molecules, so the antibody mobilization could be performed during thermal polymerization that give us more ragged devices, more ragged sensors. They are hydrophilic, so we have to dope the membrane with calcium ions to form microdomains for the nanobodies to attach. The antibody loading increased 47%, detection limit decreased by 60%, and sensitivity increased by 13%. That were good news for us. But the relative standard deviation gave us a wider range. That we had a problem with the noise. Background noise increased 3 times. A threshold was observed for the first time for concentrations up to 3.5 nanograms per mil. If we have the threshold, we have completely different mechanism, and that's something that we had to see. Possibly we are talking about membrane spanning. The noise was too much to go to any good results, and the model fitted was actually not the antibody-based model, but the receptor-based model. So we did some more thinking, and we changed our transducer. We moved on to zinc oxide nanowalls. Zinc oxide nanowalls provide alternating layers of positive and negative ions along with the non-polar plane that create an inherent signal amplification mechanism, and they seem to be more fit to nanobodies, more suitable for nanobodies. Now we have a greatly enhanced detectability, 10 times lower detection limit than conventional antibodies, 4 times lower detectability than graphene-modified sensors. Sensor reliability was greatly compromised. We still have increased noise levels. Reproducibility decreased below 6%, but at a wider range. And unfortunately, we have 30 to 50% capture activity loss during regeneration. We do not think it's the nanobodies problem. We think it's our models and our techniques problem. So some conclusions here. We have preliminary results that are very promising for biosensor nanobody platform construction. Immobilization definitely needs a new look. We need fine-tuning. We need favorable kinetics, favorable orientation. We have to go to different lipid mixtures and evaluate the suitability. We need different and hopefully most harsh sensor reversibility means that we have to investigate. And we need a better quality control in nanobody performance just to reduce material variability. Just a few notes. We are a large research group in Greece. We have the National Technical University of Athens, the University of Athens, and the lab of biomedical engineering, who is going to do some imaging for us and some differential scanning calorimetry on the way to see how nanobodies interact with membrane fluidity and packing. And I would like to thank you all for your attention. If you have any questions, I'll be happy to answer.
Thank you. We have a couple of minutes for questions. Just from practical point of view, I wanted to know how much time do you think would take to do an assay with this biosensor, and can this be developed as a point of care, a mobile and easily done, or does it require some supporting equipment?
That's a long, bitter story. I still remember the artificial biosensor pancreas that had to be on the market since early 1980s. I don't know, nanotechnology has boosted biosensors, definitely. So there are many companies that are trying to produce lipid membrane stuff. I see a shifting in protein studies using freely suspended bilayer lipid membranes lately. I've seen some promising results in using biosensor, lipid membrane biosensor technology for drug membrane permeability studies. It depends. It could be 2 years, it could be 20 years. Engineering is not a good marriage with biosensors at the moment.
Good, thank you.